Color is sensed by comparing signals from cones with different spectral sensitivities. This video will talk about color opponency, specifically blue-yellow opponency. The blue-yellow opponent system is used to compare short and long wavelength light. When a retinal field composed of short, medium, and long cones receives short wavelength light, roughly within this range, this causes S cones to be hyperpolarized, while M and L cones remain in their inactivated depolarized state. The hyperpolarization of the S cone from the short wavelength of light it receives inhibits release of glutamate. Horizontal cells, which are continuously activated by the still depolarized M and L cones, further inhibits glutamate release. The S cone sends its output to a specialized on-bipolar cell. The glutamate receptors on the on-bipolar cell are metabotropic, and so less glutamate excites the on-bipolar cell. This excited signal is sent to a small bistratified retinal ganglion cell. This cell receives information from the on-bipolar cell and so is excited by the short wavelength light. When the system receives long wavelength light, roughly within this range, S cones are not hyperpolarized, but M and L cones are. The horizontal cells are less excited because the M and L cones are hyperpolarized, and so they do not inhibit the S cone either. As a consequence, more glutamate is released from the S cones, the bipolar cell is inhibited, and the retinal ganglion cell receives those inhibition signals and is therefore inhibited by long wavelength light. So these specialized retinal ganglion cells are excited by short wavelength light, or blue light, and inhibited by long wavelength light, or yellow light. This particular mechanism is part of how humans and other mammals perceive color. But remember, all light is colorless, and color isn't real outside of the processing systems of our brain.